Okay. Um, Hello everyone. Welcome to the second meeting for the USC Neuroscience High School Journalism Club. And this is the first meeting that we are actually going over a piece of scientific literature. Um, One thing I want to mention before going into the presentation today is I was kind of debating between two different ways to do these meetings. One way, so I'm gonna have these slideshows with all the resource, like basically the summary of the entire article on it. And what I was thinking of, one way would be to run through the slideshow and go over the entire paper, and then have a discussion at the end. And then the other thing I was thinking was going through each section, like the intro, the methodology, the discussion. And then after each section, we could have a mini discussion. And then after the entire thing, we could have a larger discussion. Today, I'm going to try running through the whole paper and then having like a big discussion time at the end. Um, but I have like for the attendance form, I have like one of the questions on the attendance form is like, which one would you prefer? So like, just let me know your feedback on that one, depending on how today's meeting goes. And also, I don't know how long the slideshow part is going to take. Like, I don't know how long it's going to take to run through my slideshow. I think the best thing would be to like have 30 minutes of running through the slideshow and then like 20 to 30 minutes of discussion time. The people who weren't able to come today submitted their own discussion questions. So we already have a lot of discussion questions, you know, on hand, and obviously y'all are the priority in the discussion. So yeah, as I go through the slideshow, just like listen, jog your memory, and yeah, get ready to talk. Hopefully. Okay, I'm gonna share my screen. 1 second. All right, can y'all see the screen? Okay. Uh, okay, I'll just start then. Um, so this week's article was Dopaminergic Mechanisms of Individual Differences in Human Effort-Based Decision Making. And like I said in that video I made, um, just from looking at the title, we can kind of see what type of article this is, what its focus is, what its main point is, and see the keywords, dopamine, um, effort-based decision making. So that's essentially what the entire article is about. Let's jump into it. So what we're going to be going over today through the slides is basically just the four parts of the article, the intro, the methods, the results, and the discussion. Then after that, we'll have our own discussion time. So for the introduction, um, this article primarily concerned around I guess the effect of dopamine on cost benefit decision making. So cost benefit decision making is exactly what it sounds like. Um, when you're given like different choices, would you rather work harder and get a bigger reward, or work less and get a smaller reward? And this is like an integral part of motivation. It's everyone experiences in life, whether you would it's even like just like I guess the most basic principle would be like study hard and get 100 on a test, study less and get a not as good grade. And it's just up to you to decide. Um, and the introduction describes a big part of previous literature, which was essentially those who were given animals that were given dopamine boosters were seen to have a greater affinity to making decisions that were higher effort based. And those who received, those who blocked dopamine, had lower affinities and wanted to do lower effort tasks. And a similar thing was observed in humans as well, especially with the drug D-amphetamine. I forget its full name. It's like Dexo something something, but I'll just refer to it as D-amphetamine. It's essentially just a dopamine booster, and it was seen that those who were given D-amphetamine, had the same similar effects as the animals, putting in higher effort to get that higher reward. So that's essentially the baseline. That's the previous literature that sets up the research that's about to come in the article. So yeah. Um, Another key concept, I guess the main concept we should go over is the neurotransmitter dopamine. Dopamine has strong ties with motivation and conditioning. So dopamine it's like a neurotransmitter, it works through the different pathways in the brain, and specifically the cortical striatal dopamine pathway is associated with motivation and learning, while s- I think the nigrostriatal pathway is associated with more movement and just like moving around. So that's why when you look at Parkinson's, like the substantia nigra, that's the part that's affected because that's like the part where it correlates with like movement. So you see the tremors and the movement disabilities in Parkinson's. But for this article, it's specifically focused on the motivational aspect of dopamine, how it regulates decisions, like I said, between cost, benefit, like this just decision making. So now we move on to the methodology. And in the methodology, there's essentially just two big things we need to look at. The EEFRT task and then PET and how they use PET in the methods. So going with the EEFRT first. The effort expenditure for rewards task, EEFRT. Like I think some people call it effort, but I'm just gonna call it EEFRT. The EEFRT task is essentially designed to assess the high versus low effort decision-making behavior in people. And it does that by manipulating the three main factors of cost-benefit decision making. The amount of effort you have to put in, high or low, the amount of reward you get, and then the probability of the reward. Something I didn't mention earlier on was the probability, and that plays a huge factor in, decision making because obviously if you're having a lower probability of getting that reward, you're not as motivated compared to when you have a higher probability of receiving the reward. And in this task, let me see if I have a slide for it. No. So in this task, I'm pretty sure you were supposed to, it was like a high or low effort, and the high effort was like the low effort was like you get your dominant hand pointer finger and then you tap for like a couple seconds and you tap on a button only a couple of times. So it's pretty easy, just tap on it. But then for the high effort, you have to use your non-dominant pinky finger and then tap really at a much faster rate for a longer time. So it's more towering, it's higher effort. And that's how they gauge the difference between high and low effort for the amount of reward. The low reward was given just set as $1, while the higher reward was set at anywhere between $1.24 to $4.30. And this range also kind of factors in with that probability aspect. So going into the probability aspect, um, each task had like I guess three different probabilities or three chances. You either had a low chance of receiving the reward, a medium chance of receiving the reward, or high chance of receiving the reward. And this percentage, like the probability of the reward, was given to you before you made your decision. So every all of these factors were given to you before you made that decision. So you knew what you were going into essentially, and you processed what the probability, what the reward, what the effort was, and then you made your own decision. And then there were 25 participants in the experiment, I believe, and then they just did the EFRT test for multiple trials, of course, to see the overarching patterns. Now moving on to positron emission tomography or PET. This is where a lot of the jargon came from in the paper because PT very sciencey, Um, but the main thing to note is that I guess I'll just go through it. A ligand known as F-phthalate was used. So the ligand is essentially just in PET. They get just like a tracer, and then it's like a biological something found naturally in your body, but then they slightly tweak it so it's radioactive. And they inject that into your body. And then since it mimics whatever's naturally found in your body, it'll just flow through your bloodstream. But then because it's slightly tweaked to be radioactive, it gives off that radiation. And then when you're placed in the PET machine, laying down, um, the sensors around you, they kind of detect where the radiation is coming from and can pinpoint where in your body that this specific tracer is moving. So it's really cool. It's really genius. It's actually crazy to think about, like, somebody actually thought of this and designed it, and it's just groundbreaking and it's incredible. And it's used like everywhere. So yeah, that's PET in a nutshell. And the specific tracer and ligand used in this research was known as F-valapride. Um, Each participant underwent two scans, a placebo scan, and then after a D-amphetamine application. So the placebo scan was to gauge, their baseline levels, and then the D-amphetamine application was obviously to engage see after boosted dopamine. And one thing they mentioned is that they didn't counterbalance, and that means that because they had 20 participants, in usual experiments, you would counterbalance. Like some of the participants would do placebo first, while others would do D-amphetamine first, but they couldn't do that, and that's for a numerous amount of reasons. Um, the first is that you could, if you had gone D-amphetamine first, you'd have kind of known that that was a D-amphetamine, and it was just like you would have to do too much statistical analysis, and it was just harder to do counterbalanced research for this specific article. So that's why they just made all 25 participants do the placebo scan first, and then the D-amphetamine application after that. Also, if you hear like people moving around, I'm sorry. It's just like all the camp people, but hopefully you don't. Okay. And then the next thing I'll go over is a non-displaceable binding potential. Um, it's essentially just, in the simplest words, it's just to see how the ligand binds, and it's just like quantitative data for that. So. As you can see here, there's equations for it. You can use this link to look into it more deeply. It's actually really interesting. But non-displaceable essentially just refers to the amount of ligand that's just not specifically binded to the specific binder you're aiming to bind it to, if that made sense. I'll go into it more in the next slide. So yeah, this should help clear things up a bit more. For the PET analysis, the researchers used a four-compartment reference tissue method, or FRTM. So this really helped me understand what non-displaceable binding potential was more. So I'll just run down through it here. And yeah, so the four compartments is the different places where the radioligand can be after it's injected in you. So for PT, after the ligand is injected, it can be in four places. It can be in the blood plasma where it's still circulating in the bloodstream. It can be a free-floating tracer where it's, in the brain, but it's not actually binded to anything. It can be non-specifically binded, which means it's binded to a receptor, but that receptor isn't the specific receptor that the researchers are looking for. Or it could be specifically binded, which means the tracer has binded to the specific receptor that the researchers are looking for. And then you get the data by comparing a reference tissue with the target tissue. So the reference tissue is where non-displaceable, the word where just like non-displacement comes from. And the reference tissue is taken from an area of the brain where researchers know that there's none of that specific receptor that they're wanting to look for is. So in this example, I think it was a cerebellum. Um, and the cerebellum didn't have any of the receptors that the researchers were looking for. So they knew that there wouldn't be any specific binding within the cerebellum. So from the cerebellum, the reference tissue was taken, and from that, they only had free traces and non-binding occur in the cerebellum because there's no specific binding. And the sum of the free tracers and the non-specific binding is what non-displaceable binding is, essentially. Compare that with the target tissue or the specific area that the researchers are wanting to analyze because they know specific binding is occurring in there, and then you get the binding potential. So that's essentially how the researchers looked at the PET. They compared the reference tissue with the target tissue and see how the specific binding area compared to the non-specific binding areas. They also used voxel-wise kinetic modeling. It's it's just like a fancy word to say that the brain was just generated into like 3D pixels. Like each voxel was like a Minecraft block, just a cube, but tiny pixels. So that instead of analyzing whole regions, the researchers would be analyzing tiny little areas within the brain. So obviously that'd be very more specific. Yeah, and then this software known as SPM5 was utilized to analyze all the voxelized data in the context of EEFRT. So SPM5 was what basically bridged the EEFRT aspect of the methodology with the PET aspect of the methodology. And it used to see like, oh, this specific participant had a lot of activity in these specific voxels of the brain. How does that correlate to the EEFRT task that they had done? And then they just made connections through that software. Okay, so that's the methodology done. Here are the results. So the researchers first investigated low probability results. So like in the EFRT, when you were given a low probability of winning the reward, it was in those tasks that the researchers focused on first. And that's because previous research has shown that there were the most differences or most changes within the brain during low probability effort-based decision-making trials. So what they found was changes, positive correlations with high effort. So within the low probability trials, they found positive correlations between activity in these areas and high individuals that put in high effort for the low probability results. And these areas were the left caudate, and the left caudate is important because it's part of the striatum, and I'll get into that more a bit later. The ventral medial prefrontal cortex, both sides, the ventrolateral prefrontal cortex, both sides, and then the inferior temporal gyrus, the left side. So yeah, those are the parts most affected by the low probability results. And you can see that a bit more here in the overall results. You can see as I don't know if y'all can see my cursor, but the 12% in the probability condition, the 12% obviously is a low probability condition. And as you can see here, the three asterisks mean it's very low p-value, so very significant. Um, and you can see all of them basically have three asterisks, meaning they have a very significant correlation with the individuals that put in high effort to the low probability conditions. And then over here, you can see the overall results for another part of the brain, the insula. And as you can see here, both the left and the right insula have some sort of significant correlation in the medium to higher probability conditions. And the interesting thing about this is that it's a negative correlation. So yeah, the main two things to take away from this is that there's a positive correlation between low effort and high, no, low probability and high effort within the left caudate, the VMPFC and the VLPFC. And then there's a negative correlation with high effort and high effort and overall probability in both the left and right insula. All right. So for the discussion, the main things, like I said, to know is that the first one is cortical striatal DA sensitivity. So the findings from the left caudate of DA sensitivity having positive correlations with high effort choices during low probability trials corroborates previous evidence, meaning that, and that previous evidence is that the striatum, which the left caudate is a part of, plays a giant role in well, the striatum's DA sensitivity plays a giant role in effort-based decision making, particularly in low probability. So yeah, this just furthermore establishes that finding from previous literature. Findings on the ventromedial prefrontal cortex and the VL prefrontal cortex are relatively new. It's not as established as striatal findings. Um, but also, previous research does indicate that these have correlations with decision making, and this research just solidifies that even more. Another key finding was that obviously the insula had opposing effects or a negative correlation with higher effort decision making. And yeah, so the more dopamine the insula got, essentially, or the more sensitive the insula got to dopamine, the less likely the individual was likely to undertake higher effort choices. And this was interesting, but it is known that the insula is usually particularly associated with punishment, and response costs and like anticipating loss or punishment. So because the insula is in charge of that aspect of decision making, honestly, it's kind of like the fear or like the scared non-risk taking aspect of decision making, then it would make sense that the insula has a negative correlation with, high effort, high risk this choices. So yeah, it's 7:25 right now. Um, Let me stop sharing because I have a couple questions from the people who couldn't attend this meeting stored up in another doc. Okay. Um, 1 second. Okay. Um, so we can do this like last time. If you have a question, just raise your hand and then I'll go down the raise hands list one by one. Or it doesn't have to be a question. It can just be something you noticed, anything you want to talk about. And then I'll try to bounce off that, give a little feedback. Other people can bounce off it too. So yeah, just raise your hand. Okay, Endo, you can go first.
One thing that I noticed or that stuck out was when you mentioned the radiation go being recognized as a part of the cell and helping change it. Would that be able to be used with other types of neurology studies? Like I know it probably will be, but with like compared to prions, would it be able to help with prion studies, you think?
Sorry, 1 second. Yeah. So your question was about like how PET could be used with prions.
Yeah.
Uh, Does anyone have any answer to that? I can try answering too. But if you have an answer, just raise your hand too.
Because based off all the research I've seen just from public access, there's no real way that people have found to deal with the prions, but the radiation you mentioned sounded interesting in a way that could relate to it.
Mm-hmm. Um, Darithi, is that an answer to this question or just a question that you have?
No, sorry, it's not an answer to this question.
Okay. What I think about, so prions is like misfolded proteins, right?
Yeah.
Um, PET is essentially right now, I think in neuroscience, in neurological studies, it's most primarily used to investigate the flow of neurotransmitters and other chemicals within the brain because it
Oh, okay.
needs to make tracers that mimic, those the chemicals or the hormones.
Mm-hmm.
In regards to proteins, I don't know. I don't have that much background knowledge on like how proteins would be used to, or how tracers would be used to like, I guess, look at proteins. I mean, prions are trackable. It's just that they're not like, it's just the problem is that, I guess the therapeutic side of prions, right? Just getting rid of them.
Mm-hmm.
Yeah. So I don't know how PET would be able to, I guess, help researchers get closer to getting rid of prions. It
All
could
right.
help track it or like locate it,
Mm-hmm.
but I don't know that much about how it would be able to, be a therapy for prions.
All right. Thank you.
Okay, Will, you can go.
Yeah, I have like a much less sophisticated statement, but something that I thought was kind of interesting, and I don't know if you can answer this, it's just something that came to mind, was if there's like genetic differences within the insula like DA sensitivity, and which would make like different like families or different genetic like codes be more willing to like work harder for a higher prize, which is kind of interesting to think that like maybe certain like families or types of people where they came from might be affected by this, and which means that they would work harder or work less hard for the reward, you know?
I actually love that question. I almost had the exact same question when I first looked into this article. And yeah, that is a very big implication from this study is that differences in these specific areas could make you, a go-getter, a successful person versus a slacker. And there's a lot of questions that arise out of that, like are you just born to be successful, are you like predetermined from birth to be a successful person or a lazy person just from your brain structure? Um, and there has and this article basically implies that that is true. People are differences in your brain, like differences in the insula or differences in your prefrontal cortex dispositionally put you where you're either a go-getter or a slacker. So yeah, I guess that's a pretty groundbreaking aspect of this specific research. Also, one thing I want to note is another dopamine-based review I looked at talked about a specific gene known as COMTD, and I think that was the name. I don't remember it quite perfectly, but it was like also about how this specific gene related to how dopamine was essentially received in the brain. So, how the dopamine would be uptaked or like how the dopamine would uptake into the receptors. And this also implies that there are genetic factors to how dopamine is regulated within the brain. So yeah, I guess your question is yes. Like the answer to your question is yes. There are genetic differences that almost, I guess, preset you to be successful. But that's a very like blunt way of looking at it. Obviously, free will and everything. But yeah, that's a very interesting thing to think about.
Got it, got it. Thank you. Thank you.
Reefa, you can go next.
Okay, so mine's kind of less of a question, but maybe just another way to think of the same thing. For like mental health conditions where they're associated with less motivation or just issues with motivation, like depression or like ADHD, could it just be that there's less dopamine and then too much insula, which is why it feels so, or why people find it so difficult to put effort, I guess?
Hmm. Um, yeah, I love these questions. So yeah, I guess looking at it from a more neuroscience rather than a psychology perspective, um, just do differences in dopamine are the reason behind, depression, lacks of dopamine, lack of serotonin. And key factors of depression is that you're less motivated. And then you tend to view yourself from a different perspective where you constantly blame yourself, where you constantly I guess you put yourself at the center when it's negative, and you do not put yourself as a part of anything positive. And like all these perspectives are affected by neurotransmitters, and that's what's so fascinating about them. It's like these chemicals are basically what your personality and your emotions are the manifestations of these chemicals. So yeah, the differences in dopamine do relate a lot with mental health issues. Another thing I want to mention is that in Parkinson's disease, like I said, in Parkinson's, it's typically correlated with like a lack of dopamine in the substantia nigra, which is the nigrostriatal pathway. So that's why there's a lot of tremors and problems with movement. But also in Parkinson's, it often comes with depression basically because there's so little dopamine in your brain and a very huge lack of motivation. And this huge lack of motivation it's like it almost blends in with that lack of movement, like where you're so demotivated that you can't even move. And it's just all connected with this specific neurotransmitter called dopamine. And this plays a huge part in mental health as well.
Okay, thanks.
Yeah. Also, I know like it's easy for me to I mean I like answering questions, but if you have any answers or if you have anything um that responds to one of these questions, then you can just blurt out. And like don't be shy. Um Shrihith, you can go next.
Okay, so my question was, so like in this experiment, like the reward was money. So how can the brain's dopamine system like respond differently when like the reward is changed for like the reward is changed to something like social approval or like praise or something?
Um, that's a really good question. Uh, I actually don't have a direct answer for this. What I'm imagining is like social approval that kind of I guess still they use money because it's like the most quantitative measurement of high versus low reward. But social reward is definitely like social praise, social reward. That's definitely something that sparks your dopamine a lot. Um, that's something really interesting to think about. Does anyone have any sort of idea or like any comment on Shreya's question?
Wouldn't it be more like person to person dependent? Because like if you value money more over like social interactions, like you're probably gonna have more dopamine when you're rewarded with like social thing versus like, if you value money more, then you're gonna be like happier if you get money.
Hmm. Yeah, well, no, that Rishi, what you said, like kind of, yeah, that does make sense because it's I guess that's another reason why they use money instead of social approval because social approval is so personal and something that's so dependent on how much you value a person. Like a social approval from your dad could be very different from social approval from a random peer in your class, you know? So, there's a reason why they didn't use it in this research, but investigating the role of social approval in dopamine would be something, I'd like to read into more, honestly. Yeah, Enzo, you could add on.
I feel like money would also be a big thing because if someone who's raised in a real who with a lot of money as a kid, find is promised to earn money from a job, they might not find it as valuable because they were raised with already having it given to them or not given to them, but they didn't view it as valuable as someone who because it's difficult to say because someone who has a lot of money could still value money, but definitely not in a different way than someone who's in severe poverty, let's say. So that could also be a big factor as well as like relate similar to the social approval, I feel.
Yeah, and just adding on to that, I feel like I'm pretty sure this was done in the US. I wonder how it would differ in different cultures. Like the US, we're very money driven.
Yeah.
We go to another place in the world, do the same thing. It might be less, more slackers, but maybe that's kind of like the norm. And go-getters aren't really like seen as such a great thing, you know?
Yeah, what y'all said, like, yeah, I definitely agree with it. Money is also, I guess, now that I look at it in this perspective, it's also really, I guess, personal too. Any sort of reward becomes personal when you look at your background, your upbringing, um, and yeah, your culture. So there's a lot of factors for this experiment. I guess they had some sort of control. Hopefully, they had some sort of control on like what type of people they brought in, like middle class, upper class, lower class. Um, but yeah, I never thought of that before. That's very interesting to think about. Shreya, you can ask your question.
Okay, yeah. So I was gonna say, like, what do you think are like the therapeutic implications of the study? Because like we can see from the study that motivation is more of like a neurochemical thing than a personality thing. So like, do you know of like any therapies like to increase like dopamine in the striatum or like another region like at the moment?
Yeah. So this is also like kind of blending neuroscience with psychology. I mean, they're always blended, but I love them both. Um, Therapies regarding like dopamine. I know this is literally just taken out of my AP Psych class, but I remember a specific therapy known as um deep brain stimulus, and rather than just dopamine, it's just them firing they're like you implant an electrode into your brain, and then the electrode stimulates like just like firing of electrical signals to your brain. So it's like basically just like an assistant for firing neurons. So I know that's one sort of therapy that affects neurons directly, not through dopamine, but like through just like deep brain stimulation. one thing I should also mention, I like to mention a lot of things, but I was listening. I don't know if y'all any know. I don't know if any of y'all know Professor Huberman. He's like a neuroscience professor at Stanford, I think. But he has a podcast, and I was listening to his podcast, and he said one of his. It was like a really interesting story where like one time I forgot why. Like he just his dopamine levels went really down in his brain, and he went to the hospital, and they gave him a drug known as L-Dopa, and then he's like, boom, he's right back. He's like, it feels really weird, but again, it's like how these chemicals are the manifest themselves into your emotions and your personality. That's really interesting. But yeah, there's obviously drugs that you can use to boost dopamine within your brain. But the problem with drugs is they're addictive. Instant dopamine, that's a big debate right now where like teenagers on social media, instant dopamine gets addictive. So there's no instantaneous therapy to, I guess my final answer would be there is chemical therapies to like dopamine deficits, but it always comes with a cost, whether that be addiction or stuff like that.
No, I also listened to that podcast like right before like I read this coincidentally,
Hmm.
and like I remember the thing about like L-Dopa. So yeah.
Awesome. Uh, Ender said something in the chat. Wait, let me look at it. Yeah, like Ender, what you're saying is like it's like expanding upon the idea of I guess the overall general idea of reward and how that's personal to your upbringing and experience and like where you live, just like every aspect of you. Um, so yeah, that's something to note. I mean, if you're really interested in it, you could do more research on it for the capstone project or just any individual project. But I'd also like to look into that more. So any of y'all, if you have like any findings on like how specifically culture and your upbringing influences dopamine and like reward, like how you perceive rewards, that'll be just send it to me. Like I'd love to look at it, and I'll try looking into that more too. Okay, is it Dolce or Dulce? You can go next.
Hi guys. Oh, chink. Someone's actually very sick.
Um, my question was how can we adjust dopamine? Because if it's too much, it may lead to addiction. So how can we regulate that?
Hmm. Um, so like too much in your brain, too much dopamine in your brain.
Yeah.
Wait, can I answer that?
Did someone say something?
Yeah, I said, can I answer that?
Yeah, yeah, yeah.
So I don't know how good of an answer like this would be, but there's like treatments like Adderall and like other things that like you can that are like prescribed and you take them in like specific amounts to like because obviously if you take a lot of it, like if you take like a lot of it or if it's like uncontrolled, then it becomes addictive. But like taking it in specific amounts based on like a prescription is like kind of one way to mitigate like the amount of dopamine levels.
Yeah. Um, I agree with what you said, like just like controlling the amount. Another thing is that dopamine it relies on like I guess like when you look at social media scrolling or like just like scrolling through TikTok or Instagram Reels, it's short term and it's constantly like you're constantly doing it, right? It's little bursts of dopamine that constantly addict you. And one thing I've know about dopamine is that after each burst of dopamine, your baseline level goes down. And after it goes down, you need time to make it go back up. But with each, like when you're addicted to something or when you're doing micro doses of dopamine, like through social media, your dopamine level consistently goes down. And that's what's leading to addiction. So, like what Bhavya said, just having controlled, I guess, slower, slower delivery of dopamine would mitigate the effects of addiction. And one other thing I think is really important to note is that you should receive dopamine after you put in work. So the best way to receive dopamine is through high effort. And that's because when you don't put in any effort and receive dopamine, your body just becomes addicted to it. But when you put in that effort, then you can't get addicted to it because you need to ha put in that effort first. So like that's why so many people promote cardiovascular exercise or like running because it's a healthy way to get dopamine within your system without becoming addicted. Okay, Kaylee, you can go next.
Okay, I don't know if there's like an obvious answer to this question, but if like dopamine and striatum like makes you work harder, but like when it's like in the insula, it makes you kind of like avoid effort. Like what happens to people who have like high dopamine responses in like both regions?
Thank you for the question. That's also something really interesting to think about. I guess what would happen is that I actually don't know. They'd like balance out. Does anyone have any sort of answer to this question? You don't have to know, just any comments.
Wait, the insula is for like perceiving loss, right? Like anticipating loss.
Yeah, it's like punishment, anticipating loss. Basically like the scaredy cat part of you where like you're just like you don't want to take that big of risks.
So maybe the response would be something like you'd still put in the effort, you'd just be more aware of the losses you could suffer if it didn't go right. That way it's kind of incorporating both parts.
Yeah, I mean that does make sense where you would be like a motivated person, but you still like understand the risks or be scared of them.
I thought like they would like cancel out, but like that makes like a lot of sense.
Yeah. Okay. If there's no one else, Shreya, you can answer. Wait, Ria, you're first. Ria, you can ask your question.
So I was just wondering like how the researchers made sure that the study participants didn't already have like pre-existing mental health conditions or like motivation issues that could skew the results in any way, like before they conducted the study.
That's a great question. I actually do have an answer to that because I think they mentioned it's like it it was in the participant sec was yeah. They don't directly mention like previous like dopamine or but they do reference like previous histories of drugs or like other hormonal effects on the body and they screen them out. So like one example is subjects were excluded if they had any history of substance abuse, current tobacco use, and use of psychostimulants more than twice in the subject's lifetime or at all in the past 6 months. So yeah, that's how they kind of screened them out. But obviously, you can't account for every instance of dopamine induction. Like you get dopamine from drinking your morning coffee. So like there wasn't a perfect way to completely screen everything out. But they got like the very big ones, and I guess they kind of filtered that out. Sure, you can ask your question.
So I know we were kind of talking about the implications that we're predisposed to being a successful person or a slacker, but I was wondering, like, considering epigenetics, the way we're brought up, like our upbringing, do you think it's possible to change the way like we genetically behave because of the way we live our life?
Before I answer this question, does anyone else have an answer to this question?
Wait, can you like repeat the question again?
Yeah, so I was just asking about like how epigenetics is an actual alteration to the DNA, just like the way the DNA is regulated in the body because of your environment and your lifestyle choices. So, considering the fact that we were talking about how we're genetically predisposed to either being successful or a slacker, is it possible to change this genetic predisposition through epigenetics?
If no one wants to go for me, I'll go, I can try answering this question. But I feel like it's a very
Um,
interesting question, so
I kind of have like a kind of sub question based off of that. Do you think do you think it'd be like uh your brain structure that determines whether you're like more successful or not? Would it be more like environmental factors? Like, if if your parents kind of have a mentality of being a slacker or go-getter,
Hmm.
that it's more likely to translate to you. And like in a lot of families, there's like breakouts that are like, either extremely a slacker or like extremely uh, you know, uh, go-getter-esque.
Hmm.
So is it is it more of a structural thing or is it more of like, what you're like that?
Good question. So I think both. Sh So your question's asking about like how if it's more biological or more about your upbringing. It's like nature versus nurture. And that kind of ties in with the epigenetics question as well. Um, for the epigenetic question, I think what I'd say is epigenetics definitely can is like once you start thinking about the genetic influence of like the genetic influence of dopamine on your brain and like how that predisposes you as a slacker or a go-getter, then like obviously that question does arise where it's like does epigenetics play a factor into that? And um this research, like the research that we read today, it's not stating that 100% your brain structure determines whether you're a slacker or a go-getter. It's just stating that these are our findings and these are the implications it has. Um, so yeah, viewing it from that implication and from that perspective, then epigenetics must have a very big impact on how like we make our decisions in our everyday life, whether it be like motivated or just a lazy decision. So epigenetics would definitely have an influence. Um, and answering Rishikesh's question, um, I feel like I guess for the nature nurture debate, this is like a if you take AP Psych, you know that this is like one of the biggest debates in psychology. Actually, so I think it's 50-50. Does anyone else have like on the overall nature nurture debate, does anyone else have any other opinions? And you can talk.
Um, I'm not sure how many studies there are, but there have been a lot of I'm not yeah, I've seen like two studies on this, not a lot, but I've seen studies of people realizing, oh, this even with the weight, I'm not sure where else I could use it. This is the first example I saw that I remember. Um, if your family was in a place that had like a big starvation, like the potato famine or something, I don't really know. Um, it was found that there were two different sides. You either crave more food, but you can't eat as much of it, or you crave less food, but you can still eat a lot. And it was really weird to notice because they saw, oh, your body thinks, oh, I need more nutrients, or it thinks, oh, or your body's like, oh, I do need more nutrients, but I can't take them all in. And I don't know if that's a neurology thing or whatever, but that's just what that reminded me of.
That's actually really interesting. Could you send that like research to me if you ever find it?
Yeah, I'll try and find it again. I read it, I think, a year ago, so I'll try and find it.
Yeah, just send it to the Discord. Um, and like what Ender said, there's like this whole thing becomes like I guess a bigger question of just like the neurological biological aspect versus I guess just um an upbringing aspect. And that upbringing aspects can be like depending on your perspective, it can be biological still using epigenetics, or the upbringing aspect can be something different, like more spiritual or like more just like mental. So yeah. Um it's just interesting to think about. I encourage y'all to think about it too because it's just a very interesting question. Does anyone else have more ideas on just overall just like nature versus nurture, biology versus upbringing?
There certain things other than like, diseases or like structural things. Like, are there certain traits that are like biological other than superficial ones or like disease prone, whatever, stuff like that?
Like you
Like
mean like
personality traits.
personality traits?
Yeah, yeah, yeah.
Uh, I mean, as you can see from this research, it's implying that you're either lazy or I guess just like saying it from like a dummy perspective, it's either implying that you're a slacker or a go-getter, depending on your biology. And so like instances like this showcase that there is a biological aspect that contributes to your personality. And that's a big part of neuroscience and psychology. Oh, sure, you can talk.
I think in my bio class we were also discussing that IQ is genetically inherited. So how smart you are is based on how smart your parents are. So that's like another trait that is genetically inherited.
Hmm. Yeah, and I think we were talking about that in my psych class too. So, um, just like discussing this, you can see that like it's not something we've completely figured out yet, you know? Researchers are still trying to see exactly to what point biology is and to what point like the other things are. And um I guess it also becomes like an ethical concern once you start researching this. But it's it's just really interesting to think about how far we're influenced by our biology and our like the science and how far we're influenced by like other people's relationships with us. But yeah, like IQ, your personality traits, they all have some sort of connections to neurotransmitters, hormones, the chemical and biological aspects of your brain. And that's something that might feel, a little depressing, I guess, at times, but it's also something really interesting to think about. And the fact that we can kind of overcome these biologies also prove that there are different aspects to who you are and what makes you up as a person. Uh, we have like 3 minutes left. But I do want to get at least one more question out. So I have like 15 questions for people who couldn't come today. Uh, I might just paste them on the Discord and then so you can just at least look at them and think about them. But I'm gonna pick one randomly, and then ask that and then see if any of y'all have an answer to that. Okay, this is by Jeffrey. Um, I guess it's very similar to what we've been discussing, but it's just asking what consequences has this study had on people with motivation issues? Would people be prescribed medication to increase their dopamine to put in more effort? So yeah, it's literally just what we've been talking about, how dopamine, how can dopamine affect your motivation? And I guess this just goes overall, it's just an overarching question that's connected to this research. So if you have any like sort of thoughts or ideas, feel free to answer.
I have something just before like we go off about dopamine. Something that's super interesting that I like tried was that like I was on my phone like all the time, like especially on social media, getting like instant dopamine. And I wouldn't say I was like fully addicted to it, but it was bad. Like I bet you guys could probably like resonate with that. What I did is I did like a 2 week like no social media detox. And it was the weirdest thing ever. Like I would enjoy eating and drinking and like going outside way more. And it's not like I was like always on my phone. But it's something to think about when you're talking about like the baseline getting reset whenever you do stuff like that. Like my baseline got raised. So I don't know. I think that we can like put all this stuff we're learning about into practice through things like that if anyone's in any interested in that.
Wait, that's awesome. Um, I remember I had to like I deleted Instagram for like a couple weeks of my AP test, and yeah, I do remember like enjoying little things more and like just enjoying life more, and that's definitely something that relates to our discussion and what we're having right now about how like dopamine relates with just how you perceive the world. So yeah, and y'all should try it too. Just like try deleting Instagram for a day, or like just try deleting Snapchat or whatever for a day, and you'll stress. And then like try focusing on like the little things like food or just the fresh air, and then you'll find yourself enjoying it a lot more. If nobody else has any extra questions, we're like exactly an hour in. So thank you so much for all coming. I apologize, Professor Winston wasn't able to make it today, and I gave her like a late heads up, but I'm pretty sure she'll be here next time. Like she confirmed everything with me. So we'll have like a professional voice in the discussion next time, hopefully. But yeah, this was an awesome discussion. A lot to think about and a lot to just, I guess, a lot to just like it revealed so many more things to explore about dopamine and neuroscience in general. So yeah, thank you for this discussion.